Fine. Let's start. Every plan to colonize Mars assumes the hard part is getting there. It isn't. The hard part is the human body, and what space does to it before you even land. Number 10. The invisible flood. Picture a bullet, now make it smaller than an atom's nucleus, strip it of its electrons, and accelerate it to nearly the speed of light. Give it enough energy to punch straight through a foot of solid aluminum without slowing down. That's a galactic cosmic ray. And between here and Mars, they're everywhere, coming from every direction, all the time. These aren't solar particles from our own sun, which are bad enough. Galactic cosmic rays come from supernovas, neutron stars, black holes, violent events in other parts of the galaxy that fling atomic nuclei across interstellar space at nearly the speed of light. By the time they reach us, Earth's magnetic field and atmosphere absorb them. We barely notice they exist. Leave Earth's magnetosphere, and they hit you. Constantly. NASA's Curiosity rover measured the radiation during its 253-day journey to Mars. It absorbed 1.8 millisieverts per day. That's roughly 75% of the average person's entire annual radiation dose every single day. A round trip for a human colonist would mean exposure of around 1,070 millisieverts total. The career limit for ISS astronauts, people who spend years training specifically for this, is between 150 and 400 millisieverts. Lifetime. So, add more shielding. That's the obvious answer. The problem is that galactic cosmic rays don't stop at shielding the way solar particles do. At these energies, when the rays hit dense material, they fragment into secondary particles, neutrons, pions, gamma rays, and those secondary particles can actually be more biologically damaging than the original ray. At some point, more shielding makes things worse. The alternative being seriously proposed is magnetic shielding, generating an artificial magnetic field around the spacecraft to deflect charged particles the way Earth's magnetic field deflects solar wind. In theory, elegant. In practice, the compact magnetic fields required for a crewed spacecraft would need to reach up to 20 Tesla. For context, the most powerful MRI machines in hospitals run at around 7 Tesla. Patients experience headaches, dizziness, and nausea from brief exposure to those fields. Nobody has studied what years of continuous exposure to 20 Tesla does to a human nervous system. The cure might be as bad as the disease, we genuinely don't know. And the colonists heading to Mars wouldn't be going for a week, they'd be going forever. Number 9. The dust that eats thyroids. Let's say we solve the radiation problem. You arrive on Mars, step out of the habitat, look at the rust-colored horizon. Beautiful, bleak, alien. And immediately, the dust gets in. Mars is covered in a compound called perchlorate. It's a salt, calcium perchlorate, potassium perchlorate, and it exists in the Martian soil at a concentration of between 0.5 and 1% by weight, everywhere. The Phoenix lander found it near the North Pole in 2008. Curiosity confirmed it at the equator. The Mars Odyssey orbiter has mapped it globally. There's no part of Mars that isn't coated in it. Now, here on Earth, perchlorate is a regulated contaminant. The safe limit in drinking water is 15 parts per billion. That's 0.000015%. Mars is sitting at 5,000 to 67,000 times that level in the soil the dust comes from. What it does to you is straightforward and not pleasant. Perchlorate competes with iodide in the thyroid gland, blocking your body's ability to produce thyroid hormones. Long-term exposure means thyroid dysfunction, metabolic disruption, fatigue, potentially severe anemia. And if you're pregnant, the effects on a developing fetus are considerably worse. But here's the thing. Martian dust is extraordinarily fine. The average particle diameter is about 3 micrometers. That's small enough to bypass the cilia in your airways, pass your body's natural filters, and settle deep in your lungs. Every time a Mars colonist removes a spacesuit in an airlock, every time the habitat door opens, that dust comes in. It clings to everything. NASA's own scientists have pointed out that it will get into everything. Into the habitat, the food, the water supply. One researcher, Dr. Peter Smith, who led the Phoenix lander mission, put it plainly. You'd better have a plan to deal with the poisons on the surface. And it gets worse. In 2017, a team of scientists published research showing that when Martian UV light, no intense, because Mars has no ozone layer, hits perchlorate in the presence of iron oxides and hydrogen peroxide, it becomes actively bactericidal, not just harmful. Lethal to microbes within minutes, the surface of Mars isn't just toxic, it's actively hostile to life in a way that seems almost deliberate. We picked the least inhospitable planet in the solar system for colonization, and it's a planetary scale chemical hazard. Number 8. The 14-year robot and what killed it. On May 30th, 2018, an orbiting spacecraft spotted the beginning of a dust storm in a region of Mars called Hellas Basin. Within 2 weeks, it had covered a quarter of the planet. By June 19th, it was officially classified as a planet-encircling dust event. The sky over Mars went dark. On June 10th, NASA's Opportunity rover sent its last transmission. The storm had coated its solar panels in dust. Without sunlight, no power. Without power, nothing. After 14 years of operating on the Martian surface, one of the most successful robotic missions in history, Opportunity went silent in 6 weeks. Engineers spent 8 months trying to wake it up. In February 2019, they declared it dead. These planet-wide storms happen roughly every 5.5 Earth years. They can last for months. They turn the Martian sky so opaque that the optical depth of the atmosphere, a measure of how much light gets through, reaches values of 5 to 10. Normal daylight conditions are around 1. Visibility goes from already dim to almost nothing. A solar-powered colony, the most practical energy source for early settlers, would experience this on schedule, repeatedly, and predictably. Power fails. Life support runs on reserves. The margins for error during months of near darkness, on a planet with no supply ships coming until the next launch window, are essentially non-existent. And in 2020, a study in the journal Science revealed something else. Those dust storms aren't just blacking out the sky, they're lofting water vapor into the upper atmosphere, where solar radiation destroys it, stripping the hydrogen and oxygen apart and letting them bleed into space. Mars is actively losing what little water it has left. Every major storm takes some of it away permanently. The planet is still dying, slowly, and occasionally it does it in months-long bursts visible from orbit. Number 7. Your eyes are changing shape right now. In 2011, NASA flight surgeon Thomas Mader and his colleagues published a paper that quietly unsettled the space medicine community. They had examined 7 astronauts who had completed long-duration missions on the International Space Station. 5 of the 7 had swelling of the optic disc. 5 had their eyeballs physically flattened. The globe of the eye had changed shape. 5 had choroidal folds, which are ripples in the tissue at the back of the eye. Several had lost near visual acuity and needed stronger glasses mid-mission. The condition is now called Spaceflight-Associated Neuroocular Syndrome, SANS. What happens is this. In microgravity, body fluids don't pool in the legs the way they do on Earth. They redistribute toward the head. This raises intracranial pressure, the pressure inside the skull, which pushes on the optic nerve, which pushes on the back of the eye, which deforms it. It's a slow, quiet structural change happening to a part of your body you rather need. 29% of astronauts on shorter missions have reported negative changes in near visual acuity. For long-duration missions, the numbers are worse. Some of the structural changes, globe flattening, choroidal folds, have persisted for years after astronauts returned to Earth. No permanent blindness has occurred in ISS astronauts yet. The word yet is doing a lot of work in that sentence. NASA rates SUNS as a high likelihood, high consequence risk for missions of 1 to 3 years and is actively developing countermeasures. Lower body negative pressure devices to pull fluid back toward the legs, nutritional supplements, monitoring protocols. None of them are proven to prevent the condition. And here's the detail that makes the official confidence feel slightly thin. All of our SANS data comes from ISS astronauts. The ISS sits inside Earth's Van Allen radiation belts, which provide meaningful protection from cosmic radiation. A Mars-bound crew would be exposed to roughly 8 times the radiation Scott Kelly received during his year in space, and radiation may independently contribute to SANS's progression. We are working from a best-case data set to prepare for a much worse scenario. We know SANS gets worse the longer you're in space. Colonists would be in space for the rest of their lives. Number 6, the bacteria are adapting. In September 2006, aboard Space Shuttle Atlantis mission STS-115, astronauts carried 3 sealed containers of Salmonella typhimurium into orbit. The bacteria were safely enclosed in 3 layers of containment. All the crew had to do was push a plunger. That dropped the Salmonella into a nutritious broth and let it grow for 24 hours in microgravity. When the samples came back to Earth, Dr. Cheryl Nickerson's team at Arizona State University infected mice with both the space-grown Salmonella and Earth-grown Salmonella from the same original culture. The space bacteria killed mice faster, at lower doses, and spread more aggressively to internal organs. The bacteria hadn't changed size or shape, but they had become measurably more deadly. The mechanism was a protein called HFQ. In microgravity, the absence of normal fluid shear, the physical drag of fluids moving past cells, triggers a global shift in gene expression across the bacteria. Hundreds of genes change their activity simultaneously, and the resulting organism is better at forming biofilms, more resistant to acid, more resistant to white blood cells, and more capable of colonizing internal tissues. Why microgravity? Because the fluid environment in space is eerily similar to the fluid environment inside the human gut. Low shear, slow moving. It appears that what microgravity does is mimic the exact conditions where Salmonella normally operates as a pathogen. The bacteria aren't adapting to space, they're adapting to us. Meanwhile, independent sampling of ISS surfaces has found that 22 of 29 strains of Staphylococcus and Enterococcus collected from the station were already resistant to at least one antibiotic. A 2024 paper in The Lancet Microbe confirmed increased virulence in multiple pathogens from ISS samples, including Aspergillus fumigatus, a fungus that attacks the lungs. NASA's own records show 29 infectious disease incidents across 106 shuttle flights. There's no hospital on Mars, there's a medical kit, whatever antibiotics they packed, and a 24-minute communications delay in each direction before anyone on Earth can even respond to say they're thinking about it. Number 5, 1 year in space, 1 year to recover, forever. Identical twins make for unusually clean science experiments. When one of them goes somewhere strange and comes back changed, you can measure exactly what changed because you have a second copy of their genetics sitting on Earth for comparison. From March 2015 to March 2016, NASA astronaut Scott Kelly spent 340 days aboard the International Space Station. His identical twin, Mark Kelly, remained on Earth. 10 independent research teams spent the next several years analyzing samples, scans, and data from both men. The results were published in the journal Science in April 2019. The headline finding that circled the internet, astronauts' DNA changed by 7%, was technically inaccurate. What changed wasn't his DNA sequence. What changed was his gene expression, how his genes were behaving, which genes were turned up or down. After 340 days in space, over 7,000 of Scott's genes had altered their activity levels compared to his twin brother on the ground. When he came home, most of those changes resolved. Most. 7% of the gene expression changes. Over 800 genes, including ones related to immune response and DNA repair, were still altered 6 months after landing, not declining toward baseline. Still altered. There were also chromosomal inversions, stretches of DNA that had physically reversed direction on the chromosome. This is a known consequence of radiation exposure. 9 months after landing, the inversions were still elevated. Susan Bailey, the radiation cancer biologist at Colorado State University who led that part of the study, said it plainly. The inversions were persistent. Scott's telomeres also behave strangely. Telomeres are the protective caps at the ends of chromosomes. They shorten as we age, and shortened telomeres are associated with cellular breakdown and disease. In space, Scott's telomeres grew longer. That sounds like good news. It isn't, necessarily. Longer telomeres in a high radiation environment are associated with more aggressive cancers. The radiation can cause telomere-lengthened cells to continue dividing when they shouldn't. 2 days after landing, most of the telomere lengthening rapidly reversed. The mechanism driving this remains unknown. Scott Kelly's ISS radiation exposure was approximately 8 times lower than what colonists would receive during a Mars transit. The body keeps a record. The question is what that record looks like after a lifetime instead of a year, at 8 times the dose, with no trip home and no baseline to return to. Number 4. Nobody has ever had a baby up there. Every human being who has ever lived was born on Earth. Every single one. Over 150 billion people across all of human history on one planet. No human has ever been conceived in space. No human has ever been born in space. We have no data on what happens to human reproduction in the conditions that would be required for a Mars colony. None. What we do have is troubling. In March 2026, researchers at Adelaide University published a study in the journal Communications Biology. They wanted to know whether human sperm could navigate toward an egg in microgravity. So, they put sperm from humans, mice, and pigs into a device that simulates weightlessness, rotating in all directions simultaneously, and ran it through tiny channels designed to mimic the female reproductive tract. On Earth, roughly 50% of human sperm successfully navigate those channels. In simulated microgravity, that number dropped to under 20%. The sperm weren't damaged. Their speed and motility were unchanged. They simply lost their sense of direction. They don't really know which way is up or down, said lead researcher Nicole McPherson. The cells that did reach the egg sometimes showed unusually promising early development, but far fewer of them got there. And fertilization is only the beginning. Embryos exposed to simulated microgravity for 24 hours after fertilization showed delayed cell division and reduced cell counts, early markers that something is going wrong at a fundamental level. In pigs, fewer fertilized eggs developed into blastocysts, the stage required for implantation. Then there's radiation. Data from therapeutic radiation research suggests that exposure to Mars transit-level radiation would destroy approximately 50% of a female astronaut's ovarian reserve, the finite pool of eggs she was born with. Not over a career. For one trip, 50% gone. Here's what makes this particularly striking. Human pregnancy is currently contraindicated during spaceflight. That's not a suggestion. It is an active policy position that spaceflight is too dangerous for a developing fetus to be exposed to. The scientists who study this topic are genuinely, openly alarmed. A 2025 review paper in the journal NPJ Microgravity identified microgravity, cosmic radiation, Martian soil dust, and social isolation as 4 independent threats to pregnancy viability, each one separately capable of derailing the process, all of them present simultaneously on Mars. Every colonization plan assumes reproduction will work itself out. Every single one. None of them specifies how. Number 3, the math is brutal. Genetics has a way of making grand ambitions feel small. You can build the rocket. You can land on the planet. You can solve radiation shielding and perchlorate filtration and food production. And then population genetics taps you on the shoulder and says, how many of you are going? A genetically viable isolated population, one that can survive for multiple generations without accumulating dangerous levels of inbreeding, without losing critical immune diversity, without being wiped out by a single disease event, requires a minimum number of people. Geneticists and conservation biologists have studied this question intensively, mostly because it applies to endangered species. The rough heuristic that's been around since 1980 is the 5,500 rule. Populations below 50 face near-term extinction from inbreeding. Populations below 500 face long-term extinction from genetic drift. But those are absolute minimums for short-term survival. For a genuinely self-sustaining colony over multiple generations, accounting for demographic accidents, disease outbreaks, the statistical near certainty of severe events over centuries, and the basic requirement that people actually be able to find genetically appropriate partners. The numbers climb. Anthropologist Cameron Smith at Portland State University ran the calculation in 2014. His conclusion was that a properly structured founding population, the right age distribution, sex ratio, genetic diversity, would need to be between 14,000 and 44,000 people. He proposed 40,000 as a safe figure, not a starting crew, a founding population. Current proposals for Mars colonization, including SpaceX's publicly stated ambitions, envision initial groups of hundreds scaling to a few thousand over years. That's not close to 14,000. And here's the compounding problem. Every generation a colony spends below the viability threshold is a generation that slightly narrows the gene pool. More recessive diseases expressing narrower immune response diversity, reduced adaptability when something new comes along. The damage isn't catastrophic and sudden, it's arithmetic, working quietly against you, every generation, for centuries. Frederic Morin at the University of Strasbourg modeled what happens with a much smaller group. 98 people, carefully selected under strictly managed breeding programs, and found they could theoretically sustain themselves across a 6,300-year journey. 98 people. But that model requires a society willing to submit to scientifically managed reproduction continuously for 6,000 years, to restrict who can have children with whom based on genetic compatibility assessments for generation after generation, presumably through technological collapses, internal conflicts, leadership changes, and the inevitable human tendency to stop following rules that feel abstract. No civilization in human history has maintained any program, institution, or social practice continuously for even 500 years without catastrophic interruption. We're asked to imagine one maintaining a genetic management program for 6,000. The math isn't just brutal, it's patient. Number 2, what you'll give your grandchildren. There's a particular quality to damage that skips a generation. It isn't immediate enough to feel like consequence. It isn't distant enough to feel theoretical. It lands precisely on the people who were never consulted. In 2000, a team led by geneticist Yuri Dubrova at the Medical Research Council in the United Kingdom published a study in the journal Nature. They had irradiated male mice, just the fathers, not the mothers, and then studied the offspring. The first generation, the children of irradiated males, showed elevated rates of genetic instability. That was expected. Radiation damages germ cells, mutations pass on. But then they looked at the second generation, the grandchildren of irradiated males who had themselves never been exposed to radiation. The instability was still elevated, sometimes more elevated than in their parents. This is called transgenerational genomic instability. The irradiated parent doesn't just pass on specific mutations, they pass on a state, a disrupted cellular environment in which the rate of new mutations is abnormally high across subsequent generations. The damage compounds itself. In 2025, a study in Scientific Reports found evidence of this same signature in humans. The children of German military radar technicians, men who were occupationally exposed to ionizing radiation decades earlier, showed distinct mutational patterns in their DNA not seen in unexposed control families. Human transgenerational effects from radiation in people. In children who were born after the exposure. Now put that alongside what a Mars colonist's body will have been through. During transit alone, exposure of around 1,070 millisieverts. Earth's average annual natural background radiation is 2.4 millisieverts. The transit exposes colonists to roughly 450 years worth of Earth radiation in 7 months, and then they live on Mars, where surface radiation without Earth's magnetic field protection continues for the rest of their lives. Their children will be born there. Their children's children will be born there. Each generation adding its own exposure to the inheritance from the one before. The grandchildren of the first colonists will have never chosen any of this. They will never have stood on Earth. They will never have been protected by the magnetic field that has shielded every human who ever lived. And their genomes will carry the accumulated record of every generation's exposure above them. The 2000 Nature study found that in some cases, the second generation instability was greater than the first. The damage wasn't diminishing, it was amplifying. We don't fully understand the mechanism, we don't know the ceiling, and the people it will affect most will be the ones with the least power to object. Number 1. The question we'll never be able to ask again. Here's the thing about Mars that nobody in the colonization conversation seems to want to look at directly. We don't know if Mars has life. Not definitively, not yet. Mars once had liquid water for a very long time in conditions we now understand were hospitable to microbial life. Whether something arose in that window, whether Mars independently developed biology before losing its atmosphere, is genuinely one of the most consequential open questions in the history of science. The answer would tell us whether life is something the universe does easily, everywhere conditions allow, or whether Earth is a fluke, a single extraordinary accident in 400 billion galaxies. The answer would reshape everything we think we know about our place in the cosmos. We haven't found it yet. but we haven't looked very hard. We've landed robots in a handful of locations on a planet the size of Earth's total landmass. We've scratched the surface, literally. We haven't drilled into the subsurface, where liquid water may still exist, where life, if it's there, would almost certainly have retreated as the surface became hostile. The scientific case for looking harder, more carefully, with better tools is overwhelming. And a human colony would end that search permanently. Humans carry approximately 100 trillion microorganisms across 10,000 species in their microbiome. These cannot be removed. Sterilizing a robot to planetary protection standards is hard but achievable. Sterilizing a human is not possible without killing them. Every time a colonist steps outside, every time an airlock cycles, every time a spacesuit seal degrades slightly in the Martian cold, Earth organisms enter the Martian environment. A 2021 analog mission, a simulation of Mars habitation conducted in a desert on Earth, found that a single simulated EVA transferred 44 bacterial generi from the habitat into the surrounding soil. 44 generi. One walk. On actual Mars, those Earth organisms would encounter a sterile or near-sterile environment, and some subset of extremophile bacteria, the kinds that survive vacuum, radiation, and desiccation, would likely persist. They would spread. And after any period of time, determining whether any organic signature found in the Martian subsurface was Earth contamination or indigenous Martian life would become essentially impossible. The most interesting thing about this, though, isn't the forward contamination risk. It's what NASA's own chief medical officer pointed out in a National Academies review. He said he was less worried about Martian microbes coming back and infecting humans than he was about the other direction. Earth microbes being carried to Mars, spending years mutating in the space radiation environment, and being carried back to Earth by returning colonists. Not alien life, our own life, changed by a place we sent it to. So here's the full picture. We want to colonize Mars to ensure the survival of humanity, to spread our species beyond a single fragile world. But the act of colonizing it would permanently compromise our ability to answer the only question that would tell us whether life needs to be protected in the first place. Whether it's rare, whether it's precious, whether Earth is an exception or a rule. Once the first human boots hit the Martian soil, that experiment runs contaminated forever. Every molecule of DNA we bring with us is a smudge on the most important piece of evidence we have ever had the chance to examine. We have been looking at Mars for a long time, with telescopes, with robots, with instruments of extraordinary sensitivity. And the planet has not yet told us what it contains. That silence might be the answer. Or it might be something we haven't looked for yet in quite the right way. The terrifying reason we may never colonize another planet isn't radiation, or poison soil, or mutating bacteria, or the math of genetic survival. It's that to get there safely and sustainably, we'd need to solve all of those things simultaneously. And to send people there at all, even one person, on one mission, is to make a choice on behalf of everyone who ever wanted to know whether we're alone. It's not a question you get to ask twice. Thank you for watching and sticking till the end. We've got plenty more videos coming in the future. Hit that subscribe button so you don't miss them. See you in the next one.